Hi teacher, I'm Alejandro Salazar and I'm going to talk about the cell and the organs. So, what is a cell? Okay, the cell is the smallest unit of life. Of life. All living things have cell. Cell works like tiny factories inside of the animals and plants and humans. I'm going to talk about two types of cells. In addition of eukaryotic and prokaryotic, I'm going to talk about the animal cells. The animal cells are in animals, including humans, and the plant cells. They are in plants like trees and flowers. So, what is inside of a cell? Okay, inside of a cell we have organelles. What's an organelle? An organelle is like a little machines that do very important jobs. And the organelles in animals and plants are the nucleus, that is basically the brain of the cell. We have the mitochondria that give energy, and we have the cell membrane. The cell membrane like protect the cell. And there are some organelles that is only in a plant, like the chloroplast. And the chloroplast basically helps to make food from the sunlight. And we have the cell wall. The cell wall gives a shape to the cell and protection to the cell. And this is all from my part. Thank you for your attention.
In the first century AD, Seneca described that by using a transparent spherical container filled with water, it was possible to survey small fine prints, large and clear. In 1021, I had published his books of optics, in which light is relative to vision and the formation of image, and the function of lens is described. In the 16th century, Dux Fair and Son pair named Hans and Charles Jensen made the first compound microscope. In 1665, cities did not yet have the cleanliness and engine machinery we have with today, so light, fields, and unchanged conditions. We are daily occurrence. London did not space his situation, exploited by one of its best scientists, Ruther Hauck, who designed a microscope made of the true lens. The instrument consisted of four tubes inside a 15 centimeters. With Robert Hooke was a pioneer in microscopy. In 1610 and 77, London, a Dutchman in desk who was making a living silk cloth, made fascinating discoveries of his one. Anton van Leeuwenhoek was a master of making living silk cloth, made fascinating discoveries of his own. The cell theory was proposed by Matthias Schilling. Robert Hood is credited with his work micrography published in 1665. Almost 200 years later, Leeuwenhoek observed the first living cells. A characteristic of cells are all living things are made of cells. The cells are complex and highly organized. Cells possess the ability to replicate. Cells obtain energy. Cells interact with the environment. The cells contain a DNA. Um, This is all.
Hi, I'm Cynthia, and I'm gonna talk about the different forms of cell transport, such as passive, active transport, osmosis, diffusion, facilitated diffusion, endocytosis, and exocytosis. For this topic, I'd like to mention that a cell is like a nightclub. The cell is an exclusive party in a club, and the substance, such as water, oxygen, and glucose, are the ones who want to enter or leave. The passive transport is the free and dry. Here, substance can enter or leave the club without using energy. It's like the doors are open and people can move freely depending on how crowded it is inside or outside. The types of passive transport, the diffusion, is the natural flow of the party. If there are more people outside than inside, they will enter until everything is balanced. One example is when oxygen enters the cell because there is more oxygen outside than inside. And the next one is the osmosis, the deep water pass. Only applies to water. Water moves from waters more to waters less until it's balanced. One example is if a cell is in salty water, it will lose water and shrivel it up like a raisin. The next one is the facilitated diffusion, the bouncer assisted in dry. Some substances need help to get in because they are too big or can't pass through on their own. One example is the glucose needs special proteins in the membrane to let it in, like a big waste on the list. The active transport is the cover change entry. Here the cell uses energy, ATP, because it moves substance against the flow, like security pushing people against the natural movement. The types of active transport is the endocytosis. Everybody gets in. The cell wraps around substance and pulls them in like a bouncer wrapping a group and pulling them inside. The cell eats battery or light particles. It's one example. The exocytosis, the next one, is everybody get out. The cell pushes substance out like security throwing out drunk people. One example is the cell expels water or proteins it makes. And that's all. Thank you for listening.
According to WHO, mothers between 19 and 15 years old have a risk factors for adolescent mothers, such as illegal abortions, physiological violence due to abandonment, child abuse, social rejection of the mother, school dropout, and multiple health problems for both the mother and her baby. Most of the case mothers face complications such as preeclampsia and eclampsia. Preeclampsia refers to the increased blood pressure during pregnancy and delivery, directly affecting the brain, kidneys, lung, and eclampsia is where seizures occur during labor, which can lead to neurological damage. Another common complication, hemorrhage, which refers to excessive uterine bleeding caused by overdimensioning during labor. I'm going to talk about the addictions. One of them are social networks. They are fundamental to keep us informed. Little mouse in the box, Skinner asked himself a question. What if sometimes foods come out and sometimes it doesn't? The algorithm by social networks is based on Skinner model, which is confirmed by three stages: motivating, signal, and skills. Motivating is a fear of social regression. Skills is a lever to pull us easier. Signal is all the notifications. Mexico, 70% have obesity. 80% of serotonin is produced by our digestive system. Most of the cases we eat so much junk food for anxiety. The challenge will be to master your brain, recognize your bad habits, and nourish your body the right way.
My name is Eugenio, and today I will talk about the characteristic of living things, difference between eukaryotic and prokaryotic cells. First of all, the prokaryotic. Well, its meaning is before the nucleus, and it can only be found in bacteria and archaeas. They are unicellular organisms, and it is said that they were the first cells in the planet to appear. They have genetic material in the cytoplasm, as the eukaryotic cells. Genetic material is part of a nucleus, but prokaryotic cells does not have a nucleus, just the genetic material. If they had a nuclear membrane surrounding the genetic material, then it will be a nucleus, but it isn't. The prokaryotic cells are surrounded by a rigid cell wall composed of epidoglycan, a molecule made up of short chains of amino acids or peptides linked to sugar molecules called polysaccharides. Now the eukaryotic cells. Its meaning is true nucleus. Eukaryotic cells are a bit more complex than the prokaryotic ones because they are fundamental units of organism and they are so varied. There are in the protists, in animals, in plants, and in fungi. So they are highly diverse. The essential difference of these two types of cells is that the eukaryotic cells does have a nucleus, where the genetic material is surrounded by a nuclear membrane that separates the genetic materials from the rest of the cytoplasm and the rest of all the organelles. The function of this nucleus is to regulate the activities that take place in the cell. Now, there are some organelles that are in these eukaryotic cells. First of all, some with membranes, as the cell membrane, the nucleus, the rough and smooth endoplasmic reticulum, the vesicles, the Golgi apparatus, the vacuoles, the mitochondria, and the chloroplast. And there are also some, but without the membranes, as the ribosome, the cytoskeleton, and the centrioles. And each one of them have a specific function that helps to all the cell to work properly. Thank you.